Good afternoon. Let's try that again. Good afternoon. It's the last day of January and we survived. Just barely in some cases. My name's Martin Garner. I'm Dean of the Kramer Family Library and I'm honored to serve as your MC for this competition. And we have 6 amazing women of science who are here to win you over with their research. Now, the 6 people that they have to really convince are sitting here in the front row. I want to take a moment to introduce our judges. And so we have Dr. Robin Marschke from Institutional Research. We have Professor Tabby Farney from the library, best place on earth. Dr. Mandy Hansen from International Office. We have our Provost Emerita. Yes, Peg Bacon. We have our Associate Vice Chancellor for Research, Jesse Smith. And for diversity's sake, we did have to have one man on the panel. And so we have our Provost, Tom Christensen. So the way that this is going to work is that we'll be calling up each of our researchers in turn to do their 3-minute presentation. The judges will have a chance to do their scoring, and I'll be up here to do a little bit of time killing so that they can focus on their work. And when they're all done, they'll put their heads down on the table so I know that you're done, okay? So that we can keep this moving. I want you to keep in mind that there is an Audience Choice Award. We'll talk about how that works at the end of the competition. So be tracking what you want to do. Unless you already came here with someone in mind. I know sometimes there are like voting and trials that happen where there's a predetermined outcome sometimes. Oh, okay. Oh, wait, this isn't the politics. All right, I'm not supposed to talk about that now. So our first contestant is Julie Hurd from psychology. So Julie, if you would come up on stage.
We see it in the news, and we see it in our communities. Marijuana availability and popularity has grown in the U.S. 33 states have some form of legalization, and in 2018, marijuana sales reached over $10 billion. Now, people may use marijuana for various reasons, but one particular reason may be post-traumatic stress disorder, or PTSD, which is a mental health disorder where someone experiences psychological distress following a traumatic event. Although PTSD may be why someone uses marijuana, it's currently unclear whether marijuana use helps or harms PTSD symptoms. Some studies have shown that PTSD symptoms decrease with use, where other studies have shown that marijuana symptoms or PTSD symptoms increase with marijuana use, particularly with increased frequency of marijuana use. Now, in order to understand these conflicting results, I'm interested in going beyond frequency of marijuana use with PTSD by understanding how a trauma survivor is capable of controlling their marijuana use during moments of emotional distress. So I surveyed 306 trauma survivors who had used marijuana within the last month, and we assessed frequency of marijuana use, PTSD symptoms, and control over marijuana use in 3 different contexts: in social situations, when marijuana was readily available, and during moments of emotional distress. Now, using hierarchical regression, I proposed that initially, increased frequency of marijuana use would predict increased PTSD symptoms. However, when the other variables were considered, I expected to see that increased control over marijuana use during moments of emotional distress would predict decreased PTSD symptoms over and above the other variables. Now, our results suggested that these hypotheses were correct, where initially, increased frequency of marijuana use predicted increased PTSD symptoms. However, when the other variables were included in the model, increased control over marijuana use during moments of emotional distress predicted decreased PTSD symptoms, where frequency of marijuana use, control in social contexts, and control when marijuana was readily available did not predict PTSD. Now, these results suggest that marijuana use alone may not strictly account for an increase or a decrease in PTSD symptoms. It might be that a survivor's capability to control their use during moments of emotional distress really relates to their trauma recovery. With the growing availability and popularity of marijuana use, it is essential that we disseminate this information so that trauma survivors can be informed related to their use and their PTSD symptoms. Thank you.
Thank you, Julie. Come on over here. So Julie, tell me a little bit about yourself. Where are you from originally? Where did you do your undergraduate work?
I'm originally from Iowa, and I did my undergrad work at UNI, and then I got my master's.
UNI, is that the
Sorry,
University
of Northern
really
Iowa.
nifty ideas? Or
Yes,
no? Okay.
that too.
Okay,
Yeah.
Northern Iowa.
And then I got my master's at Arizona State University.
Okay, so what brought you to UCCS? Why did you choose us for your continuing your work?
Particularly the PhD program here with a trauma emphasis. That's really where my research has been, and I want to continue that direction.
What got you interested in this topic?
I've worked with a variety of trauma populations. I started out with more sexual assault and domestic violence. I've worked in a VA setting, and I actually worked abroad in Greece for a month with asylum seekers as well.
Okay. So when you're not solving the world's problems, how do you like to spend your time?
Oh, man. Netflix.
Netflix?
Yeah, traveling too.
Okay.
I like to travel, especially to just anywhere that's not conference related sometimes, which is nice.
So what was your most recent favorite trip, and what is your most recent favorite Netflix binge?
Ooh, probably you for Netflix.
Okay.
And probably favorite trip would be Amsterdam.
Amsterdam. What brought you to Amsterdam?
What was that, sorry?
What brought you to Amsterdam?
Just to go see the sights. I mean, the culture there, the food, and my boyfriend actually had been there before and recommended that we go.
I wasn't sure if maybe because of the availability of
No,
marijuana.
yeah, I know. It is
in
funny that it
Amsterdam,
relates.
You don't have to
yes.
travel abroad, know,
No,
because
I
we
know,
are here in
Colorado.
Colorado. I know. Another reason to choose our fine programs. So I think that our judges are done. So Julie, thank you so much. Let's hear it for Julie. And we have our next finalists who will be making their case for their research is coming up to the stage. So please welcome Brittini Hill from Biology.
Last month, on a plane ride to Spain, I sat next to an Indigenous woman from the Northwest Territory in Canada. She was traveling to Madrid to speak at the UN Climate Change COP conference, to speak on behalf of her people and their lands. Thawing permafrost is causing the lakes where reindeer water to disappear, affecting hunting grounds and threatening her tribe's livelihood. Now, this is just one example of how climate change affects the environment, people, and animals. A study of 4,000 species showed that half of all species' home ranges are shifting, an untold story of climate change. On land, species are moving at a rate of 10 miles per decade, while in the ocean, this is occurring 4 times faster, and their underwater lives make them even more challenging to study. For this reason, scientists have recommended placing priority on marine species for which there is long-term data available. So this is where my work comes in. Since 1985, the Wild Dolphin Project has monitored 4 generations of free-ranging dolphins in the Bahamas, making it the longest-running underwater dolphin project in the world. Then, in 2013, imagine our shock when the dolphins we had studied on Little Bahama Bank for 28 years were nowhere to be found. After a month of searching on our research vessel, we did find the dolphins, 160 kilometers to the south on Great Bahama Bank, already home to 2 resident communities of dolphins, both bottlenose and spotted dolphins. This marked an unprecedented mass emigration event in cetaceans, the group that includes both whales and dolphins. I am working with the Wild Dolphin Project to collect data and to analyze the dolphins' new home range. This emigration event occurred after a significant decrease in both the sea surface temperature as well as the concentration of chlorophyll A, indicating a decrease in the prey availability. This study presents a unique and rare opportunity to study a marine species for which there's long-term data available. In fact, over 3 decades of data available. And to see how new interactions play out between the resident and the immigrant dolphins. As we confront global climate change, understanding a dramatic population redistribution in one community of dolphins will help to inform our conservation of less studied and vulnerable marine species. And perhaps it will help to protect the livelihood of the people whose lives depend on our oceans. And considering that every other breath we take comes from the ocean, I'd say that's all of us, wouldn't you? Thank you.
Thanks, Brittini. Thank you, Brittini. So, I think the questions are coming. Where are you from? Where did you go to your undergraduate? Other schools that you've attended?
Yes, I'm from Pittsburgh, Pennsylvania, and I got my undergraduate degree at Wright State University in Ohio.
Okay, and so I'm thinking, if I want to research dolphins, what school in Colorado would position me well for that kind of marine biology? What brought you here?
So it wasn't the mountain, and it certainly wasn't the marine mammals in Colorado. It was military orders, in fact. So my husband's in the Air Force, so I was brought here for that. But found a good compromise in taking classes here in the great biology department, but spending my summers living on the research boat in the Bahamas.
That's a pretty sweet compromise. Okay, so live in Colorado during ski season, attend a world-class university, and then summering in the Bahamas. Okay, so what was it that drew you to this research in the first place? Because again, Pittsburgh known for its dolphin populations in the Allegheny, right? So.
Yeah, there are no dolphins in the Allegheny or Monongahela rivers, but since I was a little kid, I've just always been interested in whales and dolphins. I actually used to dress up as a marine biologist and subject my friends and family to listening to my typewritten reports that I would make up. So now I'm doing it for real, and yeah.
I just started listening to this podcast called Mortified, and it's where people share their diaries and journals from when they were in middle school, and I think you may have been featured on a recent episode.
The other option, I asked my mom, the other thing I said I wanted to be when I grew up, and that was a WNBA star, and I'm unfortunately not tall enough for that, so dolphins.
I think that barriers are artificial because, really if you wanted to reach that high, I think you could. All right, thank you so much, Brittini. Brittini Hill,
Thank
everyone.
You.
All right, our next finalist coming from physics is physics. I see the physics crowd all right here. This is good. Okay, please welcome Brianne McGrath.
I have this meme stored on my computer, as well as my class lectures, vacation photos, and videos of my kids. All of this is stored using microscopic magnets. But as this meme says, something is wrong with my magnet. Let's look at a normal magnet. Grab it, put it near a paperclip, and it'll pick up the paperclip. Now, if I heat up that magnet, the paperclip will fall. We can understand this behavior by noting that a magnet is made up of atoms. Each atom acts like a tiny magnet. We use arrows to represent these little magnets. When the arrows point in the same direction, the material is magnetic. But as it heats up, the atoms start wiggling around, the arrows point in all different directions, canceling each other out, making the entire material non-magnetic. This is how 90% of magnets behave. As they heat up, they lose their magnetic properties. But my magnet does just the opposite. The material I'm working with is iron rhodium. When it is cold, the arrows point opposite to each other. It's not magnetic. And then as it heats up, the system suddenly flips and the arrows point in the same direction. It becomes magnetic. This strange behavior can be seen down here. At low temperatures, iron rhodium has no magnetization. And then as the temperature increases, the system transitions to become magnetic. This behavior is quite strange, and it is not well understood why it happens. In my research, I created a model to understand this. I looked at the atoms close by each other. Let's call them neighbors. Typically, nearest neighbors, seen here in purple with respect to the star, are the only significant interactions. But in order to explain what was going on, I had to look at an additional, more complicated 32-term interaction involving the next nearest neighbors as well, seen in green.
I took all of these ideas and more and put them into a mathematical calculation, leading to a 4,000-line computer code that takes into account the temperature, applied magnetic field, thickness of the material, and many more contributions. This model provides a physical description of iron rhodium and an insight as to why and how this transition happens. It can make predictions, such as what temperature the transition will happen at if we say make the material thinner. But why do I care to do this? This model will allow for other scientists and engineers to make use of iron rhodium in magnetic data storage. For example, the hard drive on your computer is made up of a magnetic material divided into tiny regions, each of which stores data. The smaller the regions, the more data we can fit. But currently, we can't make the regions smaller. Iron rhodium's ability to so easily manipulate its magnetic state will permit for smaller regions and a huge increase in data storage capacity. And my theoretical calculations gives the insight necessary to make these developments possible, allowing for phenomenal improvements in magnetic technology. Thank you.
Thanks, Brianne.
You're welcome.
So, what do you think my first question is?
What am I interested in?
This is not a memory
Patterns?
game. This is
No.
fine. So where are you from? Where'd you go to
I'm
school?
from St. Louis, Missouri.
Okay.
I went to school at Texas Christian University in Fort Worth, Texas. Go Frogs. And that's where I got my undergrad, and then I got my master's degree here at UCCS.
Excellent. So, what first attracted you to working with magnets?
My nearest neighbors. No.
Oh, the purples. That's great.
The purple ones, yeah. So the honest story to that is UCCS did, because my husband's military, we got orders just like Brittini, we're here, and I love physics, and I wanted to continue studying, and the magnetics department is awesome here. I got involved with some amazing advisors, and I went from not knowing I had interest to realizing how interested I am in these magnets,
So where do you hope that your work will take you next?
I don't know. My work, I never think about where my work's going to take me, because my husband's work is where we always go.
I think we need to have a longer conversation about that.
Yeah, I know.
Maybe
right?
off the stage.
Yeah. No, I really want to become a professor. I love teaching. I love sharing my love of physics with other people. So that's my main goals. So,
Okay.
yeah.
So when you're not solving problems for that other researchers can use to help us with storage, which is a huge thing.
A huge thing, yeah.
How and you have a family that you're also intimately involved with. How else do you like to spend your time?
Yeah, so I have a 2-year-old and a 4-year-old, so my time, I don't know what that is.
Okay. That is, yes. Okay. So we all hope that you can recover some time coming up soon. Thank you very much, Brianne. All right, our next finalist is making her way to the stage. So from Biochemistry, please welcome Sara Rodriguez.
Cancer. It's a powerful word that evokes strong emotions and feelings from us, like fear, sadness, and death. But what about it scares us so much? Part of it may be that we don't fully understand cancer. But the good thing is that there's research going on all across the globe every day, looking for new information to help us better understand it in search of the elusive cure. And so today, I hope to share with you a snapshot of my work and how I hope it will contribute to these efforts. I look at a mutation called a translocation. And while there is already a lot of work, but due to the cost and the complications of cancer, I do not study it in human cancer line, but in a fungus called Neurospora crassa. The benefit of using this fungus is that it allows me to study this mutation in isolation, which improves investigation. Now, there is a lot of work on translocations already, but it's still not clear what their role in cancers may be. And so if we look at the top left corner of this screen, a translocation occurs when a large portion of DNA, indicated by the zigzag green lines, is broken off from one region of the genome or chromosome and erroneously repaired or replaced into another region or portion of the genome. What's of interest for us is the interactions that occur between neighboring DNA highlighted here by the black curved arrow, and how that may be disrupted by the mutation, along with different interactions that may occur at the new location. We're also looking at the way that this mutation may also influence global genome organization in the nucleus, highlighted here in the large black circle, and long-distance interactions that may change, such as those between different chromosomes, highlighted in the black boxes. Collectively, we're looking to understand how these changes may influence the way that an organism produces proteins and cells. If we look at the theoretical data presented in these charts, a normal non-diseased organism may have a particular production of cells or proteins that may result in this outcome. However, an abnormal or diseased organism may have a very vastly different production of proteins or cells as compared to the normal, which may influence the cell production that may be a result of changes to the growth rate in that particular organism. And so ultimately, what I'm trying to understand and hope to contribute is how translocations work and what their role is in cancer. And so that ultimately, that my addition to the work of cancer that is going on right now may change the cancer narrative to include survival, triumph, and life. Thank you.
Thank you, Sara.
Thank you.
So, where are you from? Where'd you go to school?
All those fun things?
All those fun things.
Awesome. I'm originally from Paris, California. I do have another undergraduate bachelor degree in religious studies, actually from the University of California. Before the Army took me around the world for a few years, and I landed in Colorado. I was very happy to come back to school. I ended up with a bachelor's in biochemistry here and decided that I liked it so much I needed to stay and continue my work here.
Excellent. So, what got you interested in this subject in particular?
It was purely accidental, actually. So I was very lucky to join the university and join a brand new lab, the Klocko lab. And we work with genomics and DNA and organization. And we came across this particular mutation, and further investigation and research into it found that maybe more work in it could be valuable for the field. And so I'm kind of by accident, but a lot of luck too.
But it sounds like you found your passion.
I
in
did,
this
and I
work.
really do
Yeah.
enjoy what I do.
Yeah. And so what path do you see for yourself in the future?
I would really hope to be able to continue in biomedical research. I got the opportunity to serve this great country through the military, and I hope to be able to serve people in a different capacity.
And in your service, when you were traveling around the world, what places did you get to see?
I got to see some beautiful deserts in Kuwait and Afghanistan, but then I also got to enjoy the beautiful culture of South Korea.
Excellent. Okay. Well, everyone, let's thank Sara for sharing her research with us. Thank you very much. And so we are now returning to psychology, and we are welcoming our next finalist, Amanda Devane. That means clap for her, people. There you go.
PTSD affects over 20 million Americans per year and costs billions of dollars to treat annually. This is a serious and costly disease. Fortunately, we have many well-researched treatments that we know work for most people. When we look at our top-of-the-line gold standard trauma treatments, what we find is that as many as 70% of people drop out after only 2 sessions, and of those who complete treatment, a quarter of them report ongoing distressing residual symptoms. The current solution is to have people complete a pre-treatment intervention to help prep them for therapy. However, this can add months to treatment time and cost thousands of extra dollars. So how do we improve our gold standard treatments so that people not only complete treatment, but experience full symptom remission without adding significant costs or time in treatment? The answer is mastery experiences. Mastery experiences are a time in a person's life in which they felt they successfully coped with a traumatic or distressing life event. Mastery experiences increase our belief in our ability to cope with our traumatic distress, what is known as coping self-efficacy. When coping self-efficacy goes up, avoidance goes down. We no longer feel the need to avoid our traumatic distress, thus making us more capable of fully engaging with our existing traumatic treatments, leading to less dropout and fewer residual symptoms. Asking people to recall previous mastery experiences prior to a trauma intervention has the potential to significantly improve treatment outcomes because it will increase coping self-efficacy, thereby reducing avoidance, resulting in improved treatment engagement. To test this idea, I'm comparing a mastery group to a control group. Those in the mastery condition will recall 3 previous mastery experiences, and those in the control condition will recall 3 previous non-mastery experiences. After the groups recall their memories, both groups will undergo the same trauma intervention and be assessed for coping self-efficacy, dropout, and residual symptoms. My study is currently ongoing, but what we have found so far is that those in the mastery condition report an immediate significant increase in coping self-efficacy and thus far have a 0% dropout rate. Even more exciting is that those in the mastery condition report an immediate significant decrease in depression, the most commonly reported residual symptom. And this is before they even begin treatment. This is just following the mastery recall. So these early results indicate promising ways to improve treatment outcomes. What I'm proposing is a way to revolutionize how we currently provide trauma treatment without increasing things by cost or time spent in treatment. We don't need to reinvent the wheel. We just need survivors to believe they are capable of coping with their traumatic distress so they can fully engage with our existing top-of-the-line treatments that we know work so that they have their best chance at a full recovery. Thank you.
Thanks, Amanda. So I think you know the drill now at this point.
Yeah, I'm from San Ramon, California, beautiful Bay Area. I did my undergrad at UC Santa Cruz, go banana slugs, and got another banana slug in the audience. And I did my master's degree at Boston University, and now I'm here.
Okay, and what brought you here?
The trauma specialty track, and I wanted to work with Chip. We both have the same theoretical orientation.
That's a great reason to come.
Yeah.
So what brought you into this work in the first place?
I've been lucky enough to be surrounded by a lot of former service members, and when they all came back, we had the big, as a country, realization that we did not know how to serve their needs when they came back. And so now that we have more research around that to support them, we still don't have, our gold standard treatments still aren't quite there yet. And so it's like, how do we get them to be where we really need them to be?
So where do you see yourself post-UCCS?
Working in a VA, probably. They offer the best opportunities to do research and clinical work and to be able to continue to serve service members.
So do you have any free time as a graduate student?
Absolutely not, but that doesn't mean I don't engage in leisure activities.
And what sort of leisure activities do you use your precious, precious time on?
I love to read, and so I usually read before bed. I know, right?
And it warms this
I
librarian's
just finished
heart.
a book called The Invisible Library about a magic library that connects multi-dimensions.
Yes, that I know that. That is
It's just so good.
It's so,
It's so good.
good. Yes. What? We're allowed to geek out about this stuff.
We're
This is
giving you a
good.
really great book recommendation.
It's fantastic.
Yeah.
Okay, so reading. Yeah,
Yeah.
I read before bed every night too. So
Yeah, it's very
and
calming.
that way I can wake up and get on with my work, get on
Yeah.
with my day. So that's good.
Absolutely.
All right, Amanda, thank you so much.
You're welcome.
Let's hear it for
Thanks,
Amanda.
guys.
And our final finalist is making her way to the stage. From biology, please welcome Julia Hilliard.
Who in here is hungry? Well, even if you're not hungry, I'm sure you can imagine that rumbling stomach or that inability to focus or maybe even the sense that you can actually taste the food you crave. Okay, with that sense of hunger, I now want you to imagine that the last meal you ate or are maybe eating is all you get from now until June. Now, we have to suspend reality a little bit since we wouldn't survive, but for the bacteria that we study in my mentor's lab, this is its reality. So the bacteria we study is called Borrelia burgdorferi. It's a spirochete, which is a class of bacteria, and it causes the most common vector-borne disease in the United States, which has more than 300,000 cases reported per year and has no current vaccine. This is Lyme disease. Now, this infection is transmitted by multiple species of tick who carry it in their gut throughout their life cycle, which is what is behind me. However, when these ticks are hatched, these are little brown gold circles on the left-hand side, they're born spirochete-free, and they don't acquire this infection until they take their first blood meal in late summer, feeding off of a small infected mammal host. Now, at this point, both the tick and the bacteria have to survive through the winter until that tick takes a second blood meal in early summer, again from a small mammal. Now they can feed from either an uninfected or an infected small mammal host, and if they feed from an uninfected small mammal host, which is what you see on the right-hand side, then it can transmit the bacteria to that mammal, creating a whole new infected mammal for those late summer ticks to feed on. This is what we call horizontal transmission, and it's how the bacteria can maintain itself in nature. Now, this early summer time point is also when humans can become an accidental blood meal for the tick, and they can acquire the infection and get Lyme disease. And this presents as a bullseye rash on the skin at the site of that tick bite. Now, that adult tick you see at the bottom is not considered important for Borrelia's life cycle because it feeds mostly on deer and larger mammals, and these are very poor hosts for the bacteria. So, we know this bacteria is very mobile throughout its life cycle, but there is one part wherein it simply has to endure. And this is the part we're really interested in and where we left off in talking about how hungry you might be. So how does it survive in the overwintering tick? Where does it get its energy from? So we know that the bacteria can regulate or turn on a certain gene that utilizes a carbon source or a food source that this bacteria doesn't normally like to use called glycerol, but it is produced in the overwintering tick. And my project specifically revolves around a small regulatory RNA. This is a novel molecule that can regulate this gene. And I'm currently working on teasing out what part of the process this small RNA is regulating, how it does this, and to what extent it can. And understanding this pathway not only furthers our scientific knowledge, but it can also contribute to controlling this disease in the wild, thereby potentially preventing the spread of Lyme disease to the human population. Thank you.
Wow, thank you, Julia. So I think you know what I'm going to ask you.
Yes.
Yes.
So I'm also from St. Louis, Missouri, and I went to St. Louis University for my undergrad. And
A good Jesuit school.
Yes, it is. Yeah, I've actually been to Catholic schools my entire life. So
Until now.
Until now.
You know
Now I'm
that-
at a public school.
See,
Yeah.
Neither of our C's count for Catholic
Count for
here.
Catholic?
No.
Yeah, thank you.
No.
I didn't know.
Okay.
So I got my undergrad in bio there, and then I also got my master's in public health there. And now I'm here.
So I'm really interested in your research because my sister has had the chance to be a blood meal 4 times.
Same, me too.
Yeah, yeah. And her most recent one was when we were there over the summer, and I got to see that bullseye rash, which was about the size.
Oh, she did get Lyme disease.
Oh no, she's 4 infections of Lyme so far.
Wow.
Yeah,
Okay.
it's fantastic. So, how soon will you be done with your research?
Ooh, well, considering this is only my second semester, you might want to give me some time.
Okay. What led you to your interest in this topic?
I've actually been interested in infectious diseases since I was in high school, but I've also had Lyme disease twice. So when I came out here and saw that Dr. Lybecker was working on it, I kind of wanted to include both my past maybe traumatic experiences with my infectious disease curiosity. So
did you ever dress up as a CDC investigator when you were in junior high?
No. I was more of a sci-fi fantasy person, so it was usually, Cleopatra or Ariel.
Okay.
The hair.
But and now, this all kind of sounds sci-fi and definitely not fantasy to me. But where do you hope this work will take you?
I have a lot of different paths I'm considering, but I ultimately want to kind of combine both the public health with the infectious disease research, but I think understanding molecular biology is crucial for that, so this master's will definitely help tie in with some of that future research, so I'm not sure.
Excellent.
We're going to look around.
That's all right. Second
My
semester.
dad's a career counselor,
Yes.
so it'll be fine.
Well, yes. Well, Julia, thank you so much.
Thank
Let's hear
you.
for Julia.
Thank you.
So that brings us to a close for our presentations. We're going to take a little bit of time for our judges to confer. This will also be your opportunity to vote in the People's Choice Award. And so how does that work? We actually found a use for QR codes. This is exciting. Okay. Sorry, that's a little librarian joke because we've known about them for a long time. So you can either go to the web address or to that QR code. If you need to come up onto the stage to read it with your phone, you can do that. And then please go ahead and do that voting now. And then once we have everything tallied, we will be back to announce our first, second place, and People's Choice winners. There's still some food in the back there. I noticed a lot of vegetables left. So it's a sad thing we didn't have anyone from nutrition doing presentations because clearly we need that. We'll be back. You may have noticed that the music turned off. That was my signal to come up here. And so I want to thank you very much for your participation in the voting today. I also want to thank our judges, some of whom it was such a hard task that they couldn't even stay. They just disappeared to the art. No, they're standing in the back. I see that. What? You didn't, one came. Who's sitting here? The librarian. Who's dependable? The librarian. All right. Okay, and so first up, we'd like to announce our People's Choice winner, and that is Brittini Hill. Please come up to the stage. And not only do you get this fabulous certificate, I just want to make sure I'm getting this right, you will also be getting a $250 prize, not cash. We don't believe in cash, but so. Now, in our second place, who will win a prize of $500, it's a name that we've just heard. It's Brittini Hill. And so in addition to the certificate and the $500, we're going to take a picture of you with a giant check coming up. It's very exciting. Now, because she won second place, she's not eligible to win first place. So Brittini is going to have to go back to her seat. So I do want to note, the judges told me that it was a very tight decision, that we had fractions of a point separating people. And as some audience members remarked to me, they had a really hard time choosing who they wanted to vote for in the People's Choice. So I want to have a big round of applause for every participant, for every finalist. But there can only be one first place winner, and that winner is Brianne McGrath. And now keep the applause coming. And again, the first place winner will be taking home $750. So we're writing those perfectly legal checks up right now. Okay, the dry erase part, don't worry about. And so we'll be taking photographs with the winners, with our backdrop over there. We also want to make sure that we capture this moment in time when our 6 Women of Science finalists came and brought research to the House. So again, let's hear for everybody. Let's thank our judges. There's like 3 cubes of cheese left, maybe some squash, and thanks again for being part of today.